Okay, hi everyone. So to quickly introduce myself, my name's Hannah Levenson. I am a research scholar at NC State University and head of the Specialty Crops Integrated Pest and Pollinator Management Laboratory. And my research focuses on understanding human impacts on the environment and then figuring out ways that we can work to mitigate those impacts. For the most part, I work in agriculture, but I do work in other land use types. So I like to say I work in human modified environments. And what I'm trying to do is understand pest issues and then figure out how we can incorporate protecting our beneficial organisms, which includes pollinators and parasitoids. Those are the main things I work on. And I do work, I'll talk about specialty crops, fruit crops here today, but I also do research in row crops, ornamental crops, wild areas, habitat areas. So anything that's human modified and may have a beneficial organism and a pest that we want to work to balance, my research probably can help address those questions. So today I'll talk about one of the main pests that I work on, which is Spotted Wing Drosophila. And I'll talk about updates to management that we have to recommend to you all, as well as how we're working to hopefully incorporate beneficial organisms into our management plans. So let me know if at any point the screen messes up, but hopefully it's looking good for everyone. And so today we'll talk about generally the identification, detection, prevention, and treatment of spotted wing. All right. So right at the beginning, I want to highlight everyone that's been involved in this work. So I'm going to be talking about research that I've directly been a part of, as well as research that others on the team have done, and research that actually started before I started working on spotted wing. So I want to go ahead at the beginning and highlight everyone and make sure we're giving everyone credit. The main chunk of the work I'll be talking about is USDA funded. And one of the main projects is this project I'm highlighting here, moving from crisis response to long-term integrated management of spotted wing Drosophila, a keystone pest of fruit crops in the United States. And this research involves researchers from 10 different states across the US. A big focus of this is to develop state-specific recommendations and crop-specific recommendations. So we have a lot of people involved, and I'll highlight areas where other people have been involved as well. And then specifically in North Carolina, I want to thank the farm owners and managers. We do a lot of on-farm work, so a lot of the research would not be possible without participation and cooperation from farm owners. I also want to thank colleagues, assistants, and undergraduates who have helped collect all of the field work that I'll be talking about in North Carolina, and then the funding sources. Okay, so with thanks out of the way, a quick overview. We'll talk about identification, life cycle, and host plants first. Then I'll go through a quick overview of control and prevention. And for the most part, this is going to be the previous research that I know about, but wasn't necessarily directly involved in, but is very relevant for North Carolina and managing spotted wing. And then I'll talk about some updated monitoring and management techniques, some of which the data has been collected in North Carolina, so it would be directly relevant to all of us. Okay, so the organism of today, spotted wing Drosophila, you can see a male and a female here. And spotted wing was first detected in the US in 2008 in California. You can see here. And then immediately spread to other parts of the country, and in a few short years was detected all the way across the country. It was found in North Carolina in 2010, and it has been here ever since. And you can see nearly every state in the US has had spotted wing detected. So they're originally from Southeast Asia, but were unintentionally introduced into the US. And even though it's been more than a decade since it's been detected in the US, they still cause millions of dollars in damages every year, and they are still a very big issue and a big pest. They're really difficult to control for many reasons, and I'll highlight a few of them now. So I want to give you a closer up view. Here is the female on the left hand side of this picture and the male on the right hand side of the picture. They get their common name, spotted wing Drosophila, from the males. They have this really distinct spot on their wings. And the reason that they are such a difficult pest to control is because the females have this very large, very strong serrated ovipositor, which you can see a zoomed in picture of here. So we do have other fruit flies in the US, and we do have other non-native fruit flies in the US, but spotted wing is unique because they have this big ovipositor that other fruit flies do not have. What that means is that they can saw through the skin on fruit, on completely sound, undamaged fruit, to lay their eggs, which other fruit flies cannot do. So spotted wing can infest totally sound fruit, where other fruit flies rely on broken or soft rotting fruit. So that's why they're a really big pest. So they lay their eggs on the fruit then hatch into larvae, and the larvae then eat the fruit basically from the inside out. And that is what causes the damages. So when fruit is infested with larvae, there's really strict tolerances for this. Right now, for the most part, if you are going to sell your fruit to market, and usually like big box stores, things that do really detailed inspections, have a zero tolerance policy for spotted wing infestation in fruit. So what that means is if they do a spot check and they find a single larva, they could reject the entire shipment of fruit, even if other fruit in that shipment do not have larvae. So this is obviously a big concern. It has really put a lot of pressure on growers. And I want to highlight really quickly that the market and that tolerance that's set is a whole nother issue and something that should be talked about and worked on, because you'll see as I go through the presentation, having 100% control of spotted wing, zero larvae in fruit, is nearly impossible, especially in the Southeast. That is not a realistic expectation. So there is this discrepancy between what's being asked of growers and what's realistic in the field. So I do want to highlight that here. But since that is the current situation, we do need to work to improve our management of spotted wing as much as we can, and see if we can come up with better management actions. So I forgot to show you this picture, but I do want to show you the difference between spotted wing and other fruit flies, just to really show you why they're such a big pest. So on the far right of your screen is the spotted wing ovipositor. It has a black arrow pointing to it, and you can compare it to the other fruit flies shown there to see just how much bigger that ovipositor is. So that's why they are in a whole different area of pest compared to other fruit flies. And I'll quickly talk about some other identification. So other than looking at the males that have the really distinct spots on their wings, you can see that with your bare eyes out in the field. The females you need to look at under the scope to really see, microscope is what I mean, to really see that ovipositor. And the other life stages also really need to be identified under the microscope. So another reason why this is a difficult pest, it's hard to identify just with everyday instruments. You do need a microscope, or a really like high level hand lens could potentially be useful. So what you can see on the left hand side is a spotted wing female at the top, and then this is an egg that's laid under the surface of a blackberry. This is a blackberry I collected in North Carolina. They are here, they're infesting our fruit. And you can see the egg just under the surface, that white like oval shape there. And then it has two spiracles, which are like the breathing tubes that stick out of the fruit. Other fruit flies have different shaped eggs, different shaped spiracles, different numbers of spiracles. So once you're under the microscope, you can learn the egg shapes and identify spotted wing that way. The larvae all kind of look the same, so it's not a really good life stage to identify, but you could look at pupa to identify. So I have several different fruit fly species here. On the far left is spotted wing in their pupal casing, and at the very top we have spiracles again. They have star-shaped spiracles, and you can compare that to the tops of the other fruit fly species, and they look different. So again, under the microscope, but once you have them under the microscope, they do look pretty distinct. So that's the level I'm going to talk about today of identification under the microscope, but I'm happy to answer questions later or give people more information later. I do want to quickly talk about the life cycle. So we have adults, males and females. They live for about 59 days. They'll mate. The females will then start laying eggs within a few days of emerging as adults and mating. They are eggs for anywhere from 12 to 3 days. After that, they hatch into larvae. They are then larvae for anywhere from 3 to 13 days. The larvae go through 3 instars before they then go into a pupa. They can be a pupa for 3 to 15 days, and then they emerge as full-grown adults again. So with all of this considered, a generation could happen between 8 to 10 days. So that means every 2 weeks, basically, a new generation of spotted wing could come out. So another reason they're really difficult to control, they have constant reproduction across nearly the whole year, depending on the weather. If it's warm enough, they're going to be out, and if there's fruit available, they're going to be laying eggs. So especially during crop harvest, every 2 weeks, you can expect to have a new generation of spotted wing. So populations build up really, really quickly. I will say this is weather dependent. They are sensitive to temperature, moisture, but if it's warm enough for insects to be out, they're probably out. They do have winter morphs, unluckily for us. So they don't necessarily go away in the winter. They go through a semi-diapause. So the winter morphs do have slow body regulation going on. They're not as active, but if there's a really warm day, they may become active again. If there is a hard freeze, that's when you're going to start knocking down the populations. So another concern is as our winters warm, we may not be knocking down populations enough to help with the next spring where these flies are then ready to become active again. The other thing to note is that flies are most active at dawn and dusk. So that's just something to keep in mind as we go through thinking about our management plans. Another reason why they're very difficult to control is because they have a very wide range of, I'm just going to say, substances that they can use to lay their eggs in. They have more than 30 different crop and non-crop hosts that they can utilize. And I'm showing just a few examples to show you just how the breadth of hosts that they could use. So you can see here we have several different flowers where spotted wings have been found in laying eggs, mushrooms, manure, also in compost. So they're not just in crop fields, they are, I would consider them to likely be ubiquitous in the landscape. So if you have compost, if you have any sort of fruiting plant, you probably have spotted wing there laying eggs. And just to show you that this is happening in North Carolina, we did a recent project with the NC State Consumer and Youth Horticulture Working Group. We collected samples across 11 different counties in North Carolina with the help of master gardeners and extension agents. In 2023, all the way through 2024, we collected monthly samples, and we just collected from anything that looked like it could host a spotted wing. And we found spotted wing emerge out of 18 different plants. This included crop, ornamental, and wild plants. We also sampled things like mushrooms, like you saw on the slide there. So it is happening in North Carolina. We are working to summarize these results, and we will share them soon. So what this means, like I said, if it's warm enough, the flies are going to be active. They have a wide range of substances that they can utilize. That means that at nearly any point in the year, they could be reproducing. So you'll see where we are right now, December. We're going to have decaying fruit from the summer. We're going to have mushrooms. They could be laying eggs in those things. Then as soon as spring starts, we have manure, we have other sources, and then that goes right into the crop season. So basically the spotted wing can be anywhere, and then as soon as our fruit is ready to harvest, they can move into those crop fields. They are there, they are in the natural areas. And so it's very hard to control because we don't really get a break from them. They can always be there, always be reproducing. And so if we narrow down to this summer season where our fruit crops are, they can utilize many different fruit crops. So the fruit crops that we consider at major risk for spotted wing infestation are strawberries, blueberries, blackberries, raspberries, cherries, and some grapes can be affected, but those are the main ones. And so what you'll see from this graph, this is data from Western North Carolina. Strawberries are early season. Spotted wing can be there, especially on warm days. As the strawberries go out of harvest, blueberries are ready. Spotted wing will move right into blueberries. As the blueberries go out of harvest, blackberries are ready and raspberries are ready. And so they can just hop from crop to crop across the entire year. And what you'll see, a lot of the work I do is in blackberries, and you'll see from this graph, what that means is that the populations have had months to build up. So for our blackberries, that is one of the most at-risk crops. It has the highest level of infestation just due to time of year, but also because as fall draws near and there's less and less resources, then the flies are going to concentrate in blackberries. So there's a lot of dynamics going on here, and blackberries have very, very high levels of infestation. And once populations grow and they get anywhere near out of control, it's really hard to recover. The other thing is spotted wing do prefer totally sound, ripe fruit, as you can see in the left-hand side. But as you can see in the right-hand side, when high population density is putting pressure on these flies, they will move to their lesser preference, the less ripe fruit. But they will and they can lay their eggs in this less ripe fruit. So basically what that means is, depending on the population density, as soon as there's fruit on a plant, it is at risk of being infested by spotted wing. So also something to keep in mind. So lots going on here. Lots of difficulties controlling spotted wing. What that means is that management in these fruit crops has been entirely disrupted now that spotted wing has been introduced. To give you an idea of just the level of disruption we're talking about. So like I said, I work a lot in blackberries. Some of you may remember before spotted wing, but blackberry growers really didn't need to apply a lot of pesticides. They maybe applied one, maybe two applications if there was a very small pest problem. Now, the main way that growers are trying to control spotted wing is weekly insecticide sprays. Depending on the variety, that could mean 8 or more sprays a season. So that is a huge, drastic increase in pesticide use, and that's happening across the entire country. So what that means is it's a huge economic burden. That's really expensive. There's environmental concerns with that much increase in pesticide use, concerns about increased worker exposure. We do have worries about pesticide resistance. I'll talk about that in a few slides. Concerns about secondary pest outbreak. I'll also explain that more. And then, of course, we need to think about our beneficials and what's going on with that. So we do have lots of concerns around this increased pesticide use. Now, like I said, one of the main tools that's still being used to control spotted wing is pesticides. So I do want to give you some information about what's even available. So all of these listed on the screen have labels to be used to control spotted wing. Now, they're not labeled for every single crop. Like, you can't use all of these in blackberries, for example. So it's crop specific, but all of these are labeled for spotted wing. And we do have a good spread of different classes of chemicals, so that's good. But what you'll notice is that this dashed line of good marks what's considered to provide good control for spotted wing in these crops. You can see that cuts our options in half basically at this point. So we don't have that many options for controlling spotted wing in these fruit crops. And I just want to give you this breakdown by crop type about some of the pesticides that are labeled for use for spotted wing in those crops, and a ranking of how well they do with control. And I want to highlight that we really do not have a lot of organic control options either. Okay, so I want to quickly go back to this and highlight two pesticides that are they're both very commonly used for controlling spotted wing. Sorry, Mustang Max and Entrust, and that is a pyrethroid and a spinosyn. And I'm highlighting them because in 2020, resistance was detected in populations of spotted wing and California strawberries for both of these pesticides, which is very unfortunate. Those are two of the main tools that growers have, which means at some point in time, they may no longer be a viable option. And there is concern now that it's been detected in California. What if this is happening in other places? So some of the project team members in 2021 and 2022 did testing across all of these different states you can see here to see if there is pesticide resistance in other populations of spotted wing. What I will say is that right now, spotted wing has only had pesticide resistance detected in California. We do not have to the level of resistance detected in other places. There's some concerns where other populations may be less susceptible to pesticides, but it hasn't reached the point of being considered resistance. But what I do want to highlight is that in California, those populations that have resistance, we have not detected that that resistance has gone away. It's actually increased in the populations. And some of the collaborators have been keeping colonies of those populations for as many as 30 generations, and the resistance is still present. So what that tells us is that once the resistance is in a population, it's not going to go away. So this is a big concern, something we definitely need to pay attention to and think about. And so in 2024, this year, we've started going back to all of these states and doing another round of testing to see if there's pesticide resistance. And I have collected flies from North Carolina. They are currently being tested to see if there's resistance. So I want to quickly talk about pesticide resistance. And this information is from our team members at UC Davis. And they are the experts here. This is not my area of expertise, but I want to really briefly introduce you to resistance. So there's a few different ways that an organism could become resistant to a chemical. The first is penetration resistance, where when in a susceptible population, they come in contact with that chemical, it's deadly. In a resistant population, when they come in contact with that chemical, it's no longer deadly. All of these are mutations that happen in the population. The second is metabolic resistance. So instead of, when the body processes a chemical, it's deadly. When a population has metabolic resistance, they're able to process the chemical and it is not deadly to them. And then the third way I'll talk about here is target site resistance. So if a chemical targets a specific function in the body, when it goes through the body and it gets to that function, in a susceptible population, it would then kill that organism. For a target site resistant population, they have a mutation where that no longer works. It no longer affects that body of function. So we do have a couple actions when each of these resistance types happen to hopefully help reduce resistance, prevent it from happening. So for penetration resistance, it really is about early detection, really monitoring these populations so that we can detect this as early as possible. For metabolic, you want to increase the ingestion of that chemical. So you could use a synergist or a bait. And for target site resistance, once that happens, you need to adjust your management program. You need to look for another mode of action. There's not a good way once that target site resistance has formed to kind of backtrack. So as far as the California populations go, testing has recently been done on what type of resistance there is. And unfortunately, different populations of spotted wing have all kinds of resistance. So depending on the chemical, depending on the population, all of these kinds of resistance have been documented. Additionally, our team has noted a new type of resistance that they didn't think was going to be happening in these populations, and that's called splicing resistance. Again, this is not my area of expertise, so I'm going to quickly explain it to you, but that's about how deep I can go. So splicing is basically like a change in gene expression related to RNA splicing, which is related to protein production. So once that mutation happens, a new variant of the protein is produced in that organism, and that new protein is resistant to the chemical. It's interesting that we had a new type of resistance in these populations, but it does mean that this is very complicated, and it's worrisome that we have all kinds of resistance happening in spotted wing. We are currently doing that 2024 testing to make sure it hasn't spread to other areas outside of California. And the big thing here to highlight is that what this means is we need to be careful with the chemicals we are applying to control spotted wing. We need to make sure that we have appropriate pesticide rotations where classes of chemicals are being changed every time you make a new application. That's going to slow down any mutations in the populations. It's going to keep them susceptible. And we'll continue to monitor to make sure it's not happening in other locations. So in addition to pesticide resistance forming, we also have other concerns with this high use of pesticides. So all insecticide types can reduce survival of beneficial organisms. This does include organic pesticides. They can also reduce their survival. So all insecticide types we need to be thinking about carefully. In addition to that, any adjuvants, any additives are also, we're getting data that they can also affect the survival of beneficial organisms. And we don't really understand fully the impacts of insecticides and adjuvants on our beneficial organisms. With that, what I mean is our predators, our natural enemies, and our pollinators. There's still a lot we don't understand, but we do know that we're seeing negative effects. So when our predators and our natural enemies are affected, this can then lead to secondary pest outbreaks. So in a typical system, those predators would be controlling these other insects. They would not be a concern, but when you knock down the predator population, those secondary pests are then not controlled anymore. So in some of these fruit crops, we do have concerns with scales, mites, and aphids because pesticide use has potentially knocked down those predators. And then, of course, we do have concerns for pollination because fruit crops do need pollination and what's going on with pollinators in this pest management. So now that we're on this topic, I want to bring up a concept of integrated pest management. There's a lot of different definitions that you could use to describe IPM. It's been around for a while, but one definition I like is it's a decision-based process involving coordinated use of multiple tactics for optimizing the control of all classes of pests in an ecologically and economically sound manner. So balancing this ecologically and economically sound manner, that's really the important part here. This is people's livelihoods, it's our food source. So we do need to make sure that we're protecting the crops, but there's all of these concerns that we know are happening. So we need to make sure that we are being environmentally conscious when we're making management decisions. So when you're thinking about IPM, you could think about it in this triangle. It's reminiscent of like the old food pyramid, where a lot of your effort focus should be at the base of the pyramid. And then as you need other tools, you can work your way up the pyramid. You should be using all of these tools in concert with each other. And then at the very top is chemical control. And in many cases, like with spotted wing, when we have a new pest that's extremely damaging, we're kind of in a crisis mode. We don't know what to do. We have a lot of concerns going on. We need to try to control this pest. A lot of times we then jump up to the top of the pyramid and start using chemicals. We understand how these chemicals are going to work. A lot of times we see immediate results. And so when we're in crisis mode, it makes sense. But we really do need to be careful when we use these chemicals, especially with the example of spotted wing and pesticide resistance. So we do need to use these other sections of the pyramid as well. And we need to start reestablishing what those management programs look like for spotted wing that include all of these different tools in addition to chemical control. The difficult part is a lot of these other pieces of the pyramid take more time to understand how they will work, where the chemical is a more immediate response. And I'll go through some examples of these different pieces of the pyramid in a second. But I also want to bring up a concept, integrated pest and pollinator management, which is what my research mostly focuses on. So one definition you could use is that it seeks to balance the management of pests alongside beneficial insects. And very similarly, you can think about it in a pyramid shape as well. It has those same tools that were listed in the other pyramid, but now there's a new base, a new step one. And that's thinking about the agro-ecosystem. So taking a more holistic approach, a more system approach, thinking about how all these things work together, thinking about what habitat is there, thinking about what crops we're even planting in a place to begin with. So adding a new first step. And then the whole idea here is that ecosystems have a lot of interactions. And when we simplify an ecosystem, that's when we start to see a lot of these negative consequences. So the idea with IPPM is preserving these multi-trophic, really complex interactions to make sure we're getting all of these functions in the ecosystem and we're protecting them. So things that we should think about moving forward when we're talking about different management options. All right, so first I'll show some of those options that are towards the base of the pyramid, the like cultural, mechanical control that we can use. And I'll quickly go through examples of each of these that you can see here. So first we have physical exclusion. So that is preventing spotted wing from ever being able to lay eggs in your fruit. And I do want to highlight that this work was done by a prior project group, Organic Spotted Wing Management. So they did exclusion trials in several different states, and they found that when the mesh is set up correctly, you're using small mesh, so less than 1 millimeter gauge, you can effectively reduce flies. And if you're doing it right, you can get 100% effective control of spotted wing. And that was done that was possible in blueberries. So you can see that this would take a lot of materials, a lot of labor to set up. So it's probably more effective for smaller scale farms, but it is a very effective option if that's something that would work in your operation. But for larger scale farms, it may not be as feasible to set up and to keep up with, but it can be effective. Another option is how you irrigate. So spotted wing can be pretty sensitive to temperature and moisture. Basically, if they prefer moisture, actually. So like after a rain event, we see an outbreak of spotted wing. And so changing how you irrigate can actually help with spotted wing infestation. And what they found here is that if you switch to drip irrigation, you can reduce the number of spotted wing that are in your fields, where overhead irrigation is basically like a rain event. It's adding a lot of moisture into that field that is beneficial for spotted wing. Another thing is mulching. So when spotted wing pupate, sometimes the pupa stay in the fruit, but sometimes the pupa drop to the ground and they pupate in the ground. So one option is if you can cover the ground, then the pupa don't have what they need to successfully pupate and emerge as adults. And what they found here is that using fabric to cover the ground under plants could be effective at reducing spotted wing infestation in the field. So that is another option. Another option is pruning. So same idea here with irrigation, that the more bushier a plant is, the more moisture it holds. Also, the more places that spotted wing can hide and avoid when pesticide applications are made. So when you prune a bush, you're exposing more of the bush to the elements. So there'll be solar radiation, there'll be less moisture. It could perhaps be a more effective spray. And so it's something to consider that if you're able to prune your bushes, it can be effective. It will depend on the crop and how your field is set up. So there's a lot of nuance there and a lot of if and ands. But if it makes sense for you, it has been shown to be effective. Another really, really big one. This is one that I think is very, very important. I want to highlight it. Harvest frequency and field sanitation are huge ways that you can work to reduce spotted wing infestation. So if you keep a really tight harvest interval, you're harvesting very frequently, that is extremely effective for reducing spotted wing, as you can see in this graph here. What you're doing is you're taking fruit out of the field, so it's not even available for spotted wing to lay eggs in. And then what that means is that there's less habitat for them to reproduce. So it can be very effective to keep up a really tight harvest interval. That does mean labor and everything associated with that, but it can be very effective. And then very closely related to this is field sanitation. So when you're harvesting, it's very important to remove any damaged, broken, overripe, rotting fruit. If you leave that in your fields or on your bush, that's just giving spotted wing a place to reproduce, and then they can immediately move into your nice harvestable fruit. So sanitation, taking out all of the fruit that's not good, you can't harvest it, you can't sell it, still remove it out of the field so that you're removing the habitat for spotted wing. So when you remove the fruit, you could leave it in a sealed container, then they can't emerge. If you leave it in direct sun, that can make them overheat. Or if you want it out of the way, you could bury it so that they just can't get out when they emerge. Another really useful tool has been cold storage. So once you harvest the fruit, this is a post-harvest control. Once you harvest the fruit, if you put the fruit in cold storage, so like a large walk-in freezer area, it can be very effective for killing the early life stages, eggs and small larvae, stopping development of larger larvae or pupa. And if you hold it at cold enough temperatures for long enough, it could kill all life stages. So we recommend holding it at 32 degrees Fahrenheit for 3 days. The sugar content in the fruit prevents the fruit from freezing, but that's cold enough to kill off the spotted wing. If this is something you're interested in, you want to look into, there is a tool, an investment analysis tool that is available for anyone to look at. If you just search NCSU SWD Blueberry Cold Storage Tool, you can find it on NC State Extension, and it is a downloadable interactive Excel spreadsheet. So you can put in the details for your operation. You can see if that expense makes sense. You can also see when your break-even point is. But we found this to be very effective, talking to the growers that use it currently. Right now, we do only have this tool for blueberries specifically, but if we get enough requests for other fruit types, we can work with our economics team to work to develop a tool for other fruit types. Okay, so I'm going to quickly go through some behavioral controls. These are not all available yet, so I won't go into detail, but just to get you thinking about what's possible to add into your management program. So the first is attract and kill. So it would attract the flies, and then it has an active ingredient. And you can see that when it's been used, it does show promise for helping with control. There's also food grade gums. So basically it is an alternative oviposition site. So instead of the flies laying eggs in fruit, they lay eggs in this gum. That gum is not a viable resource for the eggs and the larvae, so they die, and then you don't have that next generation of flies forming. And you can see that this also shows a lot of promise. Another thing that's being evaluated is an attractant called Combi Protect. Basically, it's an adjuvant. You mix it in your tank with the pesticide. You could then potentially use less volume of pesticide because it's attracting the flies to the droplets that are sprayed. And you can see that when it's been used in this graph here, there are some promising results. And then I don't have a lot of information about this, so I'll move quickly, but I did want to throw out that it's something that people are thinking about. We're also thinking about repellent. So just repelling the flies, not so they're not as attracted to the fields of fruit. But this is still being developed. But the point I want to make is that no matter what you do, understanding the infestation levels in your field is really critical. So many growers right now, if they're spraying, are using that weekly calendar basis, but they are spraying without actually knowing what level of infestation is in their field. So monitoring is extremely important so that you can apply an informed decision to your management plan. There's been a couple different ways of monitoring, and I'll go through some now. So some of the early monitoring recommendations were to monitor spotted wing adults. Now the traps have changed over time. Some people used yeast and sugar. There's a lot of different traps. The one I'm showing here is the trap I mainly use. It's a scented lure in this Tupperware takeout container, and it has RV fluid at the bottom, 50% RV fluid, 50% water. That collects the flies. They're attracted to the lure. They go in the holes in the container, and we just hang that in the fruit fields and we monitor. But a lot of other insects get in these traps. You can get high numbers of flies in these traps, and so it can be pretty time consuming to process this. You also then can't just look in the field and get an identification of if you have spotted wing or not. You really need to bring it back and process it and probably look under the microscope. So because it's been pretty time sensitive, we've actually received feedback from growers that they're not really monitoring for adults anymore, which then means they're defaulting back to weekly sprays, not informed decision making. So a lot of the effort in the past few years has been to come up with better, improved monitoring tools that actually are effective and usable by growers. So one option was, what if we used sticky traps? The idea here is you don't need to process it like the insects that are collected in the liquid. The male spotted wing are pretty easy to identify with the spots on their wings. So you could look at the sticky card with your bare eye, or you could use a hand lens and you could get a count of if there's male spotted wing. So we wanted to see if this could be used as an effective monitoring tool. So with several states in blueberries and blackberries, we set up sticky cards in fields. We monitored them for several weeks during harvest to see, are we collecting adult flies on here? Are the adult flies we're collecting on these sticky cards in any way related to the larval infestation in the fruit? And what we found is that In some states, we see this really nice relationship where the counts of males on the sticky cards are related to the spotted wing infestation in the fruit. But in other states, especially North Carolina, we are not finding any clear pattern between the male counts on the sticky cards and the larval infestation in the fruit. And that has happened with a lot of adult monitoring. We're not able to get this clear relationship between adult numbers and larval numbers. Sometimes there are adults flying in the field, there are no larvae in the fruit. Sometimes there's a lot of adults and there's not a comparable amount of larvae in the fruit. So I haven't really been able to make sense of adult counts with being able to anticipate larval infestation in the fruit. And so instead, we are developing other monitoring techniques that actually look at the larvae in the fruit. If you want to know what your infestation level in your fruit is, maybe just look in your fruit. So this is the filter method or the salt wash method. I may use both names. And I want to highlight that this work has really been spearheaded by Steve Van Timmeren. So the idea here is you do put out adult traps. You need to know when the flies start flying. How long after the winter into the spring are they becoming active in your crop field? Once they're active and you have fruit that's starting to ripen on your bush, you would then start sampling it to see if there's larvae in your fruit. So the process for sampling larvae is you collect a sample of berries. I can give you recommendations for different fruit types on how many berries we recommend, but it changes by fruit type. So once you collect that sample, you then soak it in salt water. You can use different amounts of salt water, but we have found that 1 cup of salt per gallon of water is a good measure to use. You then let it soak anywhere from 30 to 60 minutes. I like using 60 minutes because at 30 minutes, the larvae are still very active. At 60 minutes, they start dying off, and so they're easier to count and look at. But basically, the salt water is putting the berries in this anaerobic environment, and the larvae are forced out of the berries. They're trying to find a different place to be, and then we collect them in the container. You would then filter everything out, get rid of the berries, and then you have the larvae that were in that fruit, and then you can look at it under the microscope. We really like this for a couple of reasons. So the first is within 30 minutes to an hour, you have a real-time estimate of the number of larvae that are in your fruit in your field. You could do that on any day. You could do it multiple times in one day if you really needed to, but you know on that day that you have larvae in your fruit or not. The other thing is that it's pretty adaptable, and a goal has been to keep it very cost effective. So these are just takeout Tupperware containers that you can use. This is a plastic funnel. This is just simple wire mesh. We're filtering it into a coffee filter. That coffee filter we get from Amazon, so anyone can buy it. You could use another coffee filter if you have a different preference. The biggest expense is going to be the microscope, but you can still get pretty good microscopes for only a couple $100, and then it's a one-time expense. We're using table salt as well. So you just get an old milk jug, fill it up with water and table salt, and then you have what you need to do this. So very accessible, very cheap materials. You can also scale it up or down depending on how many samples you need. You just collect how many berries you want to process. What we found, though, is that this process will change depending on the fruit type. So what we're doing now is we're working to optimize the protocol to be the most effective for each fruit type. So at this point, we've optimized these steps here in the process: how many fruit you collect, how long you soak it, how you process it, how you rinse it. We've optimized that to be what we think is the most effective for detecting larvae in blueberries, blackberries, raspberries, strawberries, and cherries. And we're currently writing that up. I hope I'll be able to share it with you soon. But basically, if you want to start using this now, a really general protocol that will be able to give you good data is if you soak in 1 cup of salt per gallon of water. So just take a milk jug, put 1 cup of salt in it, fill it up with water. And then make sure that when you pour it over your fruit, you're submerging the fruit. and then soak it for 30 to 60 minutes. Like I said, I prefer 60 minutes. And then we recommend having a filter with this mesh inlay in. That way it catches your fruit and all the larvae filter down. I just rinse it in the sink with tap water. It rinses into this filter. And so we recommend using 10 mesh for the mesh in that funnel if you're going to use it. But basically the biggest thing is once you start sampling a certain way, process all your samples the same way to make sure that they're comparable across weeks and you can see how the population is changing. So hopefully that information will come out soon for you all. Okay, so the next thing is now that we have this method, we're using it in our research. This is how we monitor for spotted wing. And what we're finding is that spotted wing do move throughout the field. So this is an example blueberry field from Michigan. They have different varieties that are harvested at different times. So spotted wing are going to move across the farm. But if you'll notice, there's also higher levels of infestation at field edges compared to middles. The numbers in these circles are how many larvae were detected in the fruit. So we wanted to understand this a little bit more because this is going to be important for creating a new monitoring plan for spotted wing. And so in several states, we sampled fruit across the entire harvest season using that salt wash filter method. We collected fruit at the field edge and the field interior. We also, under the microscope, counted the eggs, and we separated the larvae into the 3 instars, first, second, and third. And what I'll show you, what we found is that we always had more infestation at the edges of the fields compared to the interior. So what that means is you can focus your monitoring efforts to the edges because they'll have earlier and higher infestation. So if your edge is infested, you can probably assume that at some point your interior will be infested. So it's important to monitor at the edge. But also, it is really important to be able to identify those smaller life stages, so eggs and first instar larvae. Because the way the life cycle works, if you're waiting until you can see the bigger life stages, the third instar larva, the pupa, that means they've already been in your fruit for at least a week. So how many smaller larvae and eggs are already there, right? You're missing a lot of the life cycle. So if you focus on the smaller, earlier life stages, we found that you can detect spotted wing infestation in the berries 2 weeks earlier, focusing on the edges and the small larvae. And that just gives you more time to respond appropriately and more time to figure out how to get the population under control. The other thing I want to just quickly throw out there, definitely in North Carolina, it also affects other states in the Southeast, is there is another fruit fly that could be in your fields. So I'm showing you Spotted Wing Drosophila on the left-hand side of the screen. What I'm showing you is larvae in those baskets after we did the filter method in the black line, and in the purple line is spotted wing counts. And you can see that towards the end of the summer, so starting in like September, the adults and the larvae are not matching up. But we have this other fruit fly, Zaprionis indianus, the African fig fly. And if you see, we have larva counts again in black, and now we have adult counts of Zaprionis in purple. Those match up much better. So these flies, we detect them in North Carolina starting in July almost every year. And by September, it seems like the population is switching in the fruit from spotted wing infestation to Zaprionis infestation. So it's just something to note. All of our management recommendations would be the same. There's nothing different we can recommend for this fruit fly, but I do want to throw it out there that there are other species that will be in the fields. And they can't lay their eggs inside the fruit like spotted wing can, but they have other behaviors that make it so that they can infest the fruit. The other thing with larval monitoring that I want to throw out there is we do have our collaborators in Washington working on AI counting of larvae. Larvae can be hard to count. It can be time consuming. So if there's a way to make this faster, that would be great. Right now, it's really good at the larger life stages, which is also what we're better at too. So we do need it to be better at the earlier life stages, but it is exciting and hopefully we could come up with something that would be helpful for you all. So now that we have this monitoring tool of using salt water to evaluate larval infestation in the fields, we want to rebuild an IPM program, rebuild management recommendations so that we're making informed decisions into a program. So for the past 2 years, we've been working on trialing this out. And the idea here is we're going to create something called a decision tree. So you sample your fruit, you do the salt wash, you get a count of how many larvae in your fruit, and then based on that number, you would then work your way through this decision tree to get to a management recommendation. So we have been trying this out for 2 years. And how we've been trying it out is we sample at 2 fields per farm in 6 different states. We have the regular spray program as the grower would do anytime. And then we have what we're calling a researcher field where every week when we monitor for larvae, we go through this decision tree and we come up with a management action. That week, it could be different than the grower field. So these 2 fields are getting different spray programs. And what we found in both years is that so far, the data is still coming in for 24, but so far in every state, we've been able to reduce by at least 1 spray, if not more, using this method. And so we're really excited about this. We're going to continue to develop it, and hopefully soon I can share in more detail with you all some of the components of this decision tree, but we want to make sure we get it right before we share it. So we have worked with our economics team to see, okay, we have this new monitoring technique. It does take time and effort and resources to do. The hope is that it's reducing sprays. What is the cost benefit of this? Is it beneficial to put in different resources and labor kind of early on to do this monitoring? So they evaluated this. So far, the data are in blueberries. And what they're finding is on the right side of this graph, the purple, that represents fruit sampling and to guide spraying and harvesting. And they are finding that that is actually giving you higher profits at the end of the day. So by having informed management, informed sprays, you're able to reduce your sprays and you're getting higher profits at the end of the year. So we are seeing really positive results of this new monitoring method. We're currently working with the economics team to do this same economic analysis with North Carolina blackberries. So we will have some blackberry data soon, but so far we've had multiple analyses in blueberries and it's looking really promising. Okay, so for the last little bit I have for the presentation, I want to kind of shift gears a little bit. We've talked about spotted wing a lot, how hard it is to control. We have some new options available that are very promising. So the other thing we need to think about is in these systems, there are beneficial organisms. And what's going on with them in terms of living in this intense pest management system. So first I'll talk about pollinators. And for this work, I think that blackberries are a really fun system, a really interesting system to ask some of these questions in. So as you can see in this photo, we'll have ripe blackberries on a bush that will be infested by spotted wing and so need to be managed. And in some varieties, at the same time, we have open blooming flowers where insects will be visiting these flowers. We presumably want insects to visit these flowers for pollination. So what does this mean for the pollinators in this system? So a first step was I wanted to know who was even in blackberries and contributing to pollination. So we did a survey of flower visiting insects in blackberry fields. We found many different kinds of insects visiting blackberry flowers. The majority was Hymenoptera. The majority of that were bees. And the majority of bees were honeybees. So the fields that I was working in, those growers do stock with honeybees, so that's not surprising. But we still did find many other wild bee species. So we found several species of bumblebees visiting blackberry flowers, several species of solitary bees, including leafcutter bees, and one I'll highlight here, Ceratina, the small carpenter bee. So while we were collecting this data, we also took measurements of berry quality as a way to estimate yield. So we collected data on the weight, length, width, height of the berries, the number of fully formed drupelets, the number of malformed drupelets, and the total number of drupelets. And then we compared that to a bunch of different insect community measures you can see on the side. And for almost every berry quality measurement, I think except for width, all the others were significantly related to wild bee abundance and total bee richness. So wild bee abundance and total bee richness is important for improved berry quality. So we do need to consider pollinators in this system because that is affecting yield. And for the other crops that are impacted by spotted wing, we also know that pollinators are important for yield in those crops as well. So something we need to consider when we're thinking about pest management programs in these crops. One other thing that's interesting about blackberries, so their branches, they're called canes. They will continuously grow through the season. So as you can see in this first picture, the fields can become pretty unruly. I'm sure some of you have experience with this. Especially the thorned varieties, that can make it very difficult. So growers will regularly go through and trim back their canes. So it's easier to navigate the fields, it's easier to manage the fields, easier to harvest. But the other thing is that when they trim these canes, they're also unintentionally creating some potential stem nesting habitat for insects. So when we were out in the fields, we noticed that we saw insect activity in these old trimmed canes. We wanted to know more about that, what's going on there. So we collected some of these old canes, brought them back to the lab, split them open, and we found many different insects nesting within these canes, including many species or several species of bees. Now, these nests don't impact the plant. For the most part, the insects are only nesting in trimmed canes that are older and dried out. They're not damaging the green growing canes. But it is interesting that we saw several different species of bees nesting in the canes. And some of these bee species we also found visiting the blackberry flowers that we found were important for berry yield. So the insects that are contributing to pollination are living in these plants that are under intense pest management every year. So the next logical question is, well, what does that mean for these nests? And so that's data we're working to figure out now. I don't have data to talk about yet. That data is still coming in for this year, but what we've done as a follow-up is we did a time series to see where and when in fields these insect nests become active. And then this past summer, I worked with an undergrad, Allison Huddleston. We collected nests at different time points in the year. We brought them back to the lab and we reared them out to see if development changes in fields with differing pesticide levels. So we collected stems from fields that did not have pesticide use, fields that had lowered pesticide use, and fields that had high pesticide use. And so now we're evaluating what that means for development, which would then impact pollinator population in these fields. So more to come on that, but it is something that we're paying attention to. And the other beneficial organism I'm going to talk about is a natural enemy, a predator of spotted wing, parasitoid wasp. And the insect I'm showing here is Ganaspis camorum. It recently had a name change. So to quickly introduce some concepts of biological control, which is what we're going to talk about here with parasitoid wasps and spotted wing. Biological control can be defined as controlling pests using other living organisms. You may have heard the term natural enemies, predators. That can all be considered biological control. There is a different kind of biological control called classical biological control, and that is controlling an invasive pest's population by importing its specialized natural enemy from its host range, its native range. And so for spotted wing, we're actually going to talk about both of these types of biological control. Now I said parasitoid wasp, so I want to make sure we all are on the same page with that. So I'll start with a parasite is an organism that lives in or off another organism, which could be called the host. But the important thing is that it does not kill the host. It needs the host to survive to get resources. A parasitoid is a parasite that eventually results in the host's death. So it does kill the host. And so when we're talking about biological control of Spotted Wing Drosophila, we have parasitoid wasps. So they do kill spotted wing that hopefully will help control spotted wing. So when spotted wing was first detected in the US, members of our group evaluated some native parasitoid species in North America to see if they could potentially help control spotted wing. So there were several different species that were evaluated, and unfortunately, it was not found that these species would provide effective control of spotted wing. And in fact, some species don't seem to parasitize spotted wing at all. So that meant that they needed to go more towards the classical biological control approach. So evaluating exotic parasitoids that were from spotted wing's native host range. So the team members did that. They evaluated 3 different kinds of parasitoid wasps from China and South Korea, which is spotted wing's native range. Of these 3, Leptopolina and Ganaspis are more common in early fruit. So that's where spotted wing is infesting fruit more so than other fruit flies because they're able to cut through the fruit skin with their ovipositor. And then Asabara is more common later in the season when there's rotted fruit. And so there's other fruit flies that are going to be around and infesting fruit at that point. And so when the team evaluated what species of flies these wasps would parasitize, not surprisingly, they found that Asabara had a wide range of species that it would parasitize, as you can see in the left side there under the name Asabara. And what that means is that would not be considered for classical biological control. It has too wide of a host range. It would never be approved. So they looked at Leptopolina. It was better, but it still will parasitize other species, so that's not great. And then they found Ganaspis, which will parasitize spotted wing and only very closely related species. And so just to summarize that in a different way, Ganaspis has a narrow host range, and then if we go up through the species, Asabara has the most broad host range. And when they evaluated these species, they found that Ganaspis and Leptopolina can successfully contribute to spotted wing control. They will parasitize spotted wing at high enough levels that it would be useful, and in multiple plant types. And because of the host range and everything else considered, Ganaspis chlorum was approved by USDA for a classical biological control agent. So starting in 2022, we did get a permit to release Ganaspis in these states listed here. Since then, the permit has been expanded to even more states. And so for the past few years, we've been evaluating if this is going to work in the US to help control spotted wing. Now I want to go over the life cycle and how these parasitoids will help control spotted wing. So if we start on the left-hand side under life cycle of Drosophila suzuki'i, you'll see that we have the adults, the male and female. The females go and lay eggs in the fruit, just like we talked about. The eggs will hatch into larvae. They'll go through 3 instars, pupate, and then emerge as more adults. But if we look in the middle, this parasitoid wasp interrupts this life cycle. So the wasp female will find a larva inside the fruit, lay her own egg inside the larva. When the fly larva goes to pupate, instead of the fly developing, the wasp develops and an adult wasp emerges. So it cuts the life cycle of spotted wing and adult wasps emerge, not adult flies. So it's preventing the population from growing when it's able to parasitize spotted wing. So just to make sure this is clear, I have a video of how this happens. So here's the wasp looking for the larva in the fruit. When she finds it, she lays her egg in the larva, the spotted wing larva. The larva will continue to develop, but when it goes to pupate, instead of the fly developing, that wasp then turns into a larva, it eats the fly, and then it emerges as an adult wasp. So adult flies will not emerge when they're parasitized. So it would be very helpful for controlling spotted wing populations if the flies can't ever emerge. So I'll show you just a summary of the different states that have been evaluating if Ganaspis will help control spotted wing. And in North Carolina, I've been working with the North Carolina Department of Agriculture. So they are building up colonies of Ganaspis. They've been releasing it at research stations so that we can evaluate, is it going to establish in North Carolina? Will it survive? Will it parasitize spotted wing? And then we can move forward once we find out if it will or not. So just to show you how small they are, here's some of the flies compared to our hands. This is the very first release of Ganaspis in 2022 on a research farm. And after releases, we evaluated to see if they would establish in several ways. So first we collected fruit. Instead of going through the salt wash method, we kept the fruit, we reared it in incubators to see if any insects emerged. And if there were parasitized spotted wing in there, the adult wasps would emerge. We also put out sentinel traps. So we put out fruit that was purposely infested with spotted wing larvae in the lab to hopefully attract parasitoids in the field to this parasitized fruit. And then we put out those liquid traps that collect adult insects to see if we could document any adult parasitoids flying around. So at first in North Carolina, at least, we weren't getting good recovery, but other states were. So I want to quickly show what other states are finding. So in total, in 2022, across the whole US, there was 51,000 Ganaspis released. And again, there was a name change. So this chart says Braziliensis, but it's now Camorum. And in 2023, there was over 110,000 released. And I don't have the numbers for 2024, but the production is increasing. So I'm expecting it to be higher. And the starred states are states that, as of 2023, had detected Ganaspis establishing. Now, I don't have all the summary for 2024 yet, but I'm very happy to report that after 3 years of releasing, in late summer 2024, we recovered the first Ganaspis camorum in North Carolina. We reared it from fruit, which means that a wasp did parasitize spotted wing, and we had a parasitized spotted wing in fruit that we brought into the lab. So it seems like it will establish. It's taking a little longer in North Carolina than some other states. But this is very exciting and it is promising, and hopefully it could be a tool to help control spotted wing. I want to talk about one other species, Leptopollina japonica. So you might remember I did just mention it, Leptopollina in the middle. It was evaluated for release, and because it can parasitize other fruit fly species, it was not approved for release. But when all of us across the US were evaluating Ganaspis, we were out in the field, we were collecting samples, we actually found that Leptopollina japonica had naturally established somehow. And it's been established pretty widely. So we've detected Leptopollina in Vancouver all the way down to Georgia. So it is all over the US now. We don't know where or how exactly it became established, but it's maybe a happy, positive coincidence because it was a species that was being evaluated for release, and now it's naturally established here. So now that we know it's here, we're looking into it, trying to understand how far spread it's established, if it will continue to persist here, and if it's going to contribute to spotted wing control. And I think it's pretty positive because what this means is we have multiple species that can be working together to lower spotted wing populations. So you'll see in this graph multiple different plant species that these parasitoids have been found in. The GB column with the green checks shows the different plant species that Ganaspis has been found in. You can see it's some, but not all. And then if you compare that to the Leptopollina column with red checks, it has been found in many more plant types. So that's very promising. It may in fact be a better predator of spotted wing than Ganaspis. The other thing is not only can these different species perhaps help control spotted wing in different plant types, maybe they can help control spotted wing at different times of the year. Maybe they can niche partition on the plant itself. Maybe some will be better at different stages of fruit ripeness. There's still a lot we don't know, but all of this is very promising and very exciting. I do want to note that there is some timing to keep in mind. So this graph shows starting in August, so it starts in the middle of the year, and it goes through till August of the next year. In blue, I'm showing spotted wing adult counts, and in the green dash line, I'm showing Leptopollina adult counts. And what you'll see is that there is a time lag between the two species. And for the most part, if you look at the 2023 time points, spotted wing is emerging a little earlier and at higher numbers than the wasps. So there is a time lag that's happening. We're finding this in other states as well. So what this means is the parasitoids are probably going to be a piece of the management puzzle. It's not like they're going to replace all these other management techniques that we have, because there's certain times of the year where they're not going to provide control. The other thing to think about is these wasp species are pretty sensitive. So we found that they're sensitive to moisture, to temperature, and they're sensitive to the pesticides that people regularly use to control spotted wing. So what this means is these parasitoid species are probably not going to do well in the crop fields where these things are being sprayed regularly, where spotted wing is hopefully the populations are being reduced in the crop fields. What's more likely is they're going to help in natural areas. And the other thing I'll show, so this is pulling data back from the decision tree project I talked about. So the gray dash line shows the grower field where we didn't change the management at all. The black line shows what we called the researcher field where we were going through the decision tree. So same farm, different fields, different management types. What I'm showing here is the spotted wing counts to show both fields had spotted wing. But they had different levels of these parasitoids at different times in the year. Now, this is just 2023 data, so we still need to process more data to see if this is just something that happened in 2023 or if this is a trend that's going to continue. But it just further highlights that we do need to think about how we're using the chemicals in this if we want to have predators, biological control, beneficial organisms in these fields as well. Our management decisions will impact these populations. So just to re-summarize, it's going to be a piece of the puzzle. So spotted wing control is going to continue to be complex. It's going to be a whole suite of different management tools that we use together at maybe different times of the year, definitely in different crops. So it's not going to be straightforward, but we're learning more and we do have good recommendations for what you can start doing and start adding on to each other. And again, I want to highlight that on the side here, we have non-crop habitats. That's going to be really important habitat for our pollinators and also for these parasitoid wasps. So like I said, we're not expecting the crop fields to be a big habitat for these wasps. But what they might do is they might help control spotted wing in these non-crop habitats. So all those other plant types that I talked about where we know spotted wing will lay eggs, maybe the parasitoids can help control populations there where we're not able to do management. Maybe they can help control overwintering spotted wing populations so that in the spring they're lower. Maybe it delays movement into crop fields. There's still a lot we need to understand, but it is a promising potential tool as long as we understand how to make it effective and how to use it and bring it into our management plans. Okay, so with that, I know I threw a lot of new information in to you. And so I thought it would be nice to just do a really quick summary of the key points. So first, even though it's been more than a decade since we found spotted wing in the US, they still cause millions of dollars in damage each year. They are probably ubiquitous in the landscape. They can utilize more than 30 different crop and non-crop hosts. And they're very difficult to control. There's lots of moving parts to keep in mind. And so still, IPM programs have not been fully reestablished. And so that's the big focus of our research is let's get IPM, maybe IPPM back out there. Let's reevaluate our management plans. Because we do have concerns for pesticide resistance forming. We do have concerns for beneficial organisms. So we need to rethink this management and make sure we're being smart. So with that in mind, we need to focus on being careful with our chemical use, make sure we're doing appropriate rotations. We're changing mode of action so that we can limit further resistance forming. Make good use of cultural control practices, use our tools that we have beyond chemicals so that we can get back to more of that triangle approach where a lot of our effort goes to the base of the triangle rather than the top. That also includes behavioral control tools. And that definitely means we need to bring monitoring back into our programs. We should know what the infestation level in our field is before we make a management decision. And so we need informed management actions. That means we need the tools to even be able to make those actions. So we are working on that. And then hopefully biological control will know more soon, and hopefully that can be incorporated into these management plans. And we also should keep pollinators in mind when we're making these decisions. So the last slide I have, and I'll leave it up maybe while we start taking questions, is we have a new survey. We want to understand what impacts people are experiencing from spotted wing infestation. It's been more than 10 years. There was a survey done when spotted wing was detected early on, but it's been a while. So we need to know what everyone's experience is, how they're being impacted. And then we also would like to know if our research is helping or if there's something else we can do. So I will put a link in the chat once I get out of my presentation for accessing this survey, but you could also scan this QR code and it'll take you to the Google form. Just to remind you that this is totally voluntary. If you start, you can stop the survey at any time and your information will be kept confidential, so don't worry about that. But that's what I had prepared, and maybe we'll keep it up while Amanda, if there's any questions, we could start taking questions.